Great. Welcome to Ask the Expert. Today we have Dr. Michael Snyder discussing personalized responses to nutrition revealed using big data and remote monitoring, coming to us from Stanford University School of Medicine. Welcome. Thank you so much for joining. And he's the Stanford Ashman Professor and Chair of Genetics and the Director of the Center of Genomics and Personalized Medicine. He received his PhD training at Caltech and carried out postdoctoral training at Stanford. Dr. Snyder has pioneered the use of big data and multiomics to develop advanced scientific discovery and transform healthcare. His laboratory has invented many technologies that are widely used in medicine and research, including methods for characterizing genomes and their products. For example, RNA-seq, NGS paired and sequencing, chip, and later chip-seq, protein arrays, machine learning for disease gene discovery, and wearables for monitoring personal health and exposures. His application of omics and wearable technologies to perform longitudinal profiling of people when they're healthy and ill is transforming medicine and healthcare. And indeed, his laboratory's recent work to use smartwatches and wearables to detect illness, including infectious diseases such as COVID-19 prior to symptom onset, is being used by many thousands of people. He's helped co-lead many large-scale projects, including ENCODE, HMP, HubMap, and H10. And he's co-founded 16 biotech companies, including Personalis, QBio, January AI, Fit-Ursine, RTMH, Marble Therapeutics, and Iolio. So thank you so much for joining us. We're really excited to hear more about your research and perhaps how it, and hopefully how it pertains to type 1 diabetes and autoimmune diabetes.
Hi, okay, great. Well, thanks very much for having me here. Yeah, as Monica said, we do a lot of work in the area of big data and health monitoring. And I thought I'd focus more generally on the topic of nutrition, where we're also using a lot of these approaches to be able to understand responses to nutrition. And I don't know how much affects type 1 diabetes, a lot affects type 2 and nutrition in general. So hopefully this will be of interest to this audience. So obviously, there's a famous saying, you are what you eat, food is medicine. I think that's something all of us would agree with. But I think even more importantly, it's not just, what you eat that makes you, it's how you respond to food. And it's a fairly relatively poorly understood area. And it's one that we've started exploring lately using some of the big data approaches that our lab is known for. So just to put this in context, we are keen on doing health monitoring using large data approaches. And so a number of years ago, it's actually about 13 now, we set up something called personal omics profiling, where we collect very, very deep data on people. And specifically, we'll sequence their genome, and then from their blood, we'll actually measure from their immune cells, peripheral blood monocyte cells, and measure their epigenome, meaning methylome, transcriptome, proteome. Out of plasma, we'll also measure proteome, cytokines, metabolites, lipids. We also follow their microbiome. This is on top of deep clinical tests, questionnaires. And we also do advanced tests on most of the folks, stress echocardiograms, also glucose control measurements like OGTT and something called SSPGE for insulin resistance. So we have a lot in this area because I actually happen to be type 2 diabetic, strange one. And then we also do a lot with wearables. So we collect a lot of very, very deep data on folks as a way of, while they're healthy, to better follow what their profiles look like. And we also do it longitudinally. So we've been running the study, over 13 years for me, and about now almost 11 years for most of this rather smallish cohorts, up to about 171 now have added on. A few have dropped off. So we're collecting these deep data to get a better picture of people's health. And then from that data collection, we actually wound up making many, many health discoveries just in the first 3.5 years. Nearly half the people, the first 109 people, 49 major health discoveries came out, meaning we actually found it spanned a wide range of areas, but we found, for example, in heme-oncology, we found some with early lymphoma, people with pre-cancers. In the area of cardiovascular disease, we found 2 people with serious heart issues, one from genome sequencing, another from wearables, and so on and so forth. So the idea was that these deep data dives, they actually give a much, much clearer picture of people's health. And I should point out, so the idea is that if your health is a 1,000-piece jigsaw puzzle, where measuring 500 or 600 pieces versus say 5 or 6 they measured today. And no one technology did this. By getting this more comprehensive view, we could follow all these things, and they were all found pre-symptomatic, I want to emphasize. So we're getting, again, a clearer picture of you before you have symptoms, and that leads to the discovery of disease early. One of the other things we discovered by doing this deep data profiling longitudinally is that we could actually see how people age. And it turns out everyone ages differently. So for example, this person in the middle, their top biochemical pathway that changes as we follow them is a cardiac hypertrophic signaling pathway. They're a cardioager, if you will. We later learned this person's stage 2 hypertensive, which sort of makes sense. This person over here, they're a fairly typical ager. They're immune, not shown here. Their metabolic pathways also increase over time. We think you need as little as 5 measurements within 2 years, and we can tell you how you're aging. And we call these aging patterns ageotypes. So each column over here is a different person. Each row is a different ageotype. And so the folks over here, these 4 folks, they're aging in all 4 of these categories: kidney, liver, metabolic, and immune. This person's a kidney ager, this kidney, metabolic, and so on. There are clinical markers associated with these. So it's very actionable information. So we've been doing this in general for health monitoring, and that's turned out to be quite powerful. But these days, we're also trying to explore the effects of nutrition on people's health. And we started this in the area of well, here's illustrating this concept. The idea is that, you know, you give a person certain foods, what is their response going to be, and how similar, how different is it? And we follow this in depth, because then we can also learn something about, you know, not only detailed response, but we learned some biochemistry, you'll see in a minute, by doing these omics and physiological profiles in high depth. And the ultimate goal is to try and understand the role of the microbiome and various other factors in your response to food. So we started this in the area of fiber, in part because fiber is known to have important health benefits. And it's, again, it sits in your colon. I'm sure that it's known to many of you. It supports your microbiome, which is in your colon mostly, well, all over your body, but especially in your colon. It's known to boost your immune system, and it clearly brings in many important nutrients. And your fiber has been implicated in actually benefits for preventing heart disease, stroke, hypertension, all kinds of areas, especially in the area of obesity and type 2 diabetes, even cancer, as many of you may know, especially colon cancer. It's very dependent on your fiber content. Now, the problem is, you hear fiber is good for you, but when you think about it, fiber is very, very heterogeneous. Short chain, long chain, hydrophobic, hydrophilic, positive, negative charge. It's all over the map. It's kind of like saying animals are all the same, you know, lumping turtles in with people and cockroaches and things like that. We would argue they're quite different. And so we're very interested in, you know, what is the effect of different fibers on people's health? And so we decided to, in a starter study, we're now running a bigger one now, but we basically analyze the effect of 2 very, very common fibers many of you may be familiar with: rabinoxylan, which is a component of physium husk and many other things, and inulin, which is also in many nuts and other areas as well. And these are very, very common fiber. If you look at the literature, they've both been reported to have health benefits, but it's been very controversial. In some cases, some studies say, oh, each of these will lower cholesterol or lower glucose, and others will say, no, that's not true, no effect. I'm not sure any said to be bad for you, but they're either reported to have no effect or have a positive effect on glucose and cholesterol. So we decided to try this ourselves in a crossover study. We had 18 participants who basically took one fiber or the other in a dose-dependent fashion. So for example, they would take 10 grams as a supplement of fiber each day, ramp it up to 20 the next week, 30 per day the next week. And they'd start with, they were randomized, either rabinoxylan or inulin. Then after that period, they would do a washout, and they'd go through the next one, and then another washout, and then they would do a mixture of those 2 fibers plus 3 others. And we would do dense sampling during this period, where we would sample them at week 0, 1, 2, 3, etc. And we would follow this out for quite some period. And then we would do our deep omics profiling that I mentioned before on these folks at each of these time points. So very, very deep data profile to see the effects of these fibers. Forgot to say, but we also do deep clinical lab testing as well. And some of the important molecules we follow, of course, are cytokines, which are inflammatory markers. So we basically ran, as I say, people through this. We saw hundreds of molecules changing, and we could, using clustering analysis, some of you may know what that means, we could actually see, the patterns of molecule change as they went through this time course. And arabinoxylan, here's two of the more interesting clusters, a few hundred molecules in each, that basically, these are the molecules that go up in response to arabinoxylan in a dose-dependent fashion. These are the molecules that go down. And there's some other patterns as well, but these are the major ones, and the ones of highest interest. And so we could see all these different changes. And what we discovered is that arabinoxylan, in general, seems to reduce cholesterol on most people. And that had been reported in literature somewhat reproducibly, but there are studies that says it has no effect. And here's arabinoxylan, here's the mixture had, less of an effect, and that might make sense. It's got less arabinoxylan. Inulin actually had no effect. We also saw, as I say, many other molecules change, and it turns out some of the bioacids, these are primary and secondary bioacids, they would actually change in response to arabinoxylan. And we think this is actually how they're thought to be metabolic products of cholesterol. So we actually think this is one of the ways, and I'll come back to this a little later, in which cholesterol gets basically removed by fiber. It gets converted into these bioacids. And inulin had no effect on cholesterol, the bioacids. We actually could even see how the microbiome shifted. There's a number of microbiome species. We did metagenomics here. And basically, these are the microbes that will actually shift. So there's arabinoxylan here, inulin here, and these are the microbes that shift in response to both. Some microbes specifically shift, excuse me, sorry, in response, thank you, in response to arabinoxylan. Other microbes will specifically respond and will shift in response to inulin. So we'll see these microbes shift in response, and we think this is important because these are obviously important for your health. Sorry, seemed to have picked up a sneeze. I won't have time to go into this, but based on all the different proteins and RNA and patterns of change, we actually could come up with a whole new metabolic pathway, not entirely new, partially new, for how we think cholesterol is actually removed. And the microbiome actually does play a part of this. So we basically could get a more systems view, if you will, of metabolism and specifically how cholesterol is changed. And one of the other things we found is that in general, although cholesterol went down in response to arabinoxylan, there were individual differences. So just to illustrate this point, this person here, number 69, they're typical. Their arabinoxylan, dropped their LDL cholesterol, and I guess inulin had no effect, as we would expect. But interestingly, in this person, arabinoxylan had no effect, and inulin actually dropped their cholesterol. So although in general there was a certain response, at the individual level, there was actually quite a bit of difference. And I suspect that's the reason the literature actually looks different, depending on the different studies that are out there, determines what is really going on. The other thing we discovered is that high doses of inulin for some people will actually induce an inflammatory response. These are cytokines that go up. This is all actually, sorry, liver one, but same with cytokines. So for 3 people, high doses of inulin will induce this. Okay. So that was one of our first forays into individual responses to different foods. So we've been doing a lot more lately with this concept of trying to do remote monitoring and healthcare, if you will, at home. And, if you think about it, these days, most people go to a physician's office to actually get routine checkups, when in reality, you could actually get a lot of health monitoring simply in your own house. And there's two approaches we've been pushing. One is the wearables, and the other is something called home microsampling, where you take little droplets of blood, mail it in, and we measure hundreds of, actually thousands of analytes. And I'll dive into that a little more deeply. I know what that sounds like, but ours actually works.
How, can I just insert a quick question there?
Yeah.
How is that microsampling at home, the adoption of it, how's that going?
Yeah, I'll tell you. So hold that question. All right. We're going to
Okay,
dive
sure.
a lot deeper. Great question, though. Sure. So on the wearables part, yeah, we'll start with that. And because I want to spend time on glucose monitoring, which I think should interest this group. So on the wearables, what we did was so there are probably know there's hundreds of devices out there, mostly in the form of watches and rings. I myself, typically wear 4 watches. One broke, so I'm down to 3, and I have my Aura ring, often a glucose monitor, and even
Wow.
hearing aids these days. Yeah, hearing aids, believe it or not, can be physiological sensors, and I actually wear some as well. I'm not wearing mine right now, but so these are powerful devices because they follow you 24/7. And this particular story, I won't dig into so deeply, but we discovered early on when we put these on our cohort, you can tell when people are getting ill from an infection. I originally discovered it when I first got Lyme, but later respiratory viral infections. We discovered you can pick those up from a simple smartwatch and a pulse ox. And so we went on again. I want to spend more time on the glucose monitoring. But we went on to show that you can actually get this published first in 2017 before COVID. And now, then COVID came out, we quickly partnered with Fitbit and showed that we could do real-time detection actually with a simple smartwatch. And so this is just one example. We enrolled 5,300 people, but we had right away, and then we had 32 who were wearing Fitbit at the time they got COVID, and they had a diagnosis day and a symptom day. And this is our very first case where this individual, this is resting heart rate. Turns out to be a very sensitive measure of illness. Actually, it's way better than temperature, especially for COVID, where a lot of people don't get the fever. But anyway, this person here, this is their diagnosis date here, symptom day on day 47. But their resting heart rate jumped up 9.5 days prior to symptom onset. So presumably they, got infected here and were possibly even spreading it around prior to getting symptoms because they had no idea. And then we went on to write a real-time detection system that actually follows your baseline in a circadian fashion. It looks for a jump up in signal. And the original, retrospectively, we can tell when you're getting COVID, a median of 4 days prior to symptom onset. Works about 80% of the time. 20% we don't get a stable baseline on folks. In the real-time detection, just because of the way we set the sensitivity, it's about 3 days prior to symptom onset, so median. And it's actually even more sensitive than an antigen assay because it's tracking you 24/7. So it can really follow in exquisite detail your physiology. So if you're interested in that, feel free to sign up for a study, and it gives you red alerts when you have a shift. Now, I should emphasize, it's not specific for COVID. Any infection will potentially trigger the way we set up with red alerts. And other things contribute.
Mike, can I just interject for
Yeah.
a second? So have you found that most sort of infectious diseases cause this sort of 9-day prior increase in heart rate, or is it variable?
It's highly variable, both with the person and with the disease. COVID has an unusually long pre-symptomatic period. So it's actually quite good for this. Things like influenza and rhinoviruses, their incubation period is more like 30 to 48 hours from the time you get infected to the time you get symptoms. So that's a shorter period. We'll still see the jump up. And again, I left that part out because I was going to spend more time on the glucose control. But we did discover that with the like influenza, we'll detect before symptoms most of the time, but it's more like 24 hours as opposed to like, this is a median of 4 days. It's not, yeah, this is For COVID, it's very easy to pick up this, but for other things, because they operate so much faster, the advanced notice is not as good. Yeah, great question though. Okay. So the area that we're spending a lot of time on these days, and I'm just gonna jump in on this, which I think should interest this crowd, is this glucose monitoring. So you may know continuous glucose monitoring has been very powerful for following type 1 and insulin-dependent type 2 diabetics. What we decided to do early on was put these on people who were so-called normal, and also pre-diabetics, just to see what their response would look like. And so what we
Pre-type
discovered,
2, pre-type 2 diabetics, right?
well, they're just normal running around people.
Okay.
Okay. So we don't know necessarily whether type 1 or type 2. In the end, some of the folks we've wound up discovering, I'll show you in a minute. Well, I won't show this part, but some are Modi, for example, folks. So anyway, just by putting these on folks, some people do have good glucose control. You know, you don't see much spiking during the day after meals. Some people are moderate spikers, and some are severe. And some of the so-called normal people, they're spiking just as severely as type 2 diabetics. Okay. So, which we did run a few of those as controls. And so we wrote algorithms, I won't get in details here, for quantifying your spiking phenotype, if you will, in your glucose, overall glucose levels. And so we can classify people in the low, moderate, or severe, if you will, glucotypes based on their glucose levels and spiking patterns. So we actually think some of these folks who are severe spikers are missed by normal tests because of the way this is done. It turns out, and this is type 2 diabetes, you may be aware, we lump everyone together, even though they may have different underlying defects. So some people, the most common one is insulin resistance, where cells don't respond to insulin, but there are many other forms as well. Some people don't make insulin who are still type 2. Some people don't release it from the pancreas. Some people have beta cell or incretin defects and so on. And so we're actually working pretty hard to subtype type 2 diabetes. I myself am type 2 diabetic. I'm insulin sensitive and I make insulin. I just don't release it from the pancreas. So I actually respond to very specific drug.
Mike, just a question from the audience. Do you have C-peptide on these subjects?
We do actually, yeah. Yeah. So
Do you share these data with others?
Yeah, we do actually. We're one of the few groups that does do this. So I wouldn't say we necessarily have it all. It depends on the study. The first study, we had it on some, but not necessarily all. And some more recent studies that we're trying to publish now, we have it on everyone. We're actually measuring their insulin resistance, their C-peptide, their incretins, and so on, and actually correlating it with their glucose response patterns. Pretty cool study. We think we can subtype the type of type 2 diabetes you have just from the glucose pattern using machine learning and other methods.
Yeah,
so
that's very valuable data set. That's great.
We think so. Yeah, thanks. And that data I didn't quite have ready for this presentation, but we think that's going to be very powerful.
Well, everyone will be looking for that paper on the horizon, I'm sure.
Okay, yeah, thanks. So anyway, we're pushing that pretty hard, and that will make then phenotyping people at home pretty easy. And these people right now are missed, the ones who are classified as normal but are severe spikers. And in general, the thought is that if you can, severe spiking is not good for you, obviously, and can lead to increased cardiovascular disease. And the thought is that these folks will become type 2 diabetic in a more classified standard classification method. So anyway, one things we discovered early on is that different people spike to different foods. And that was also found by Aaron Siegel's group around the same time. And this is some work from Aaron's group, but some people, just to illustrate this, will spike to potatoes, others to pasta, some to white bread, others to brown bread. It's all over the map, and it's very, very personal. And this is a picture from Aaron's paper. This is an individual who spikes a banana, but not a cookie. Probably the only person on the planet doesn't spike to a cookie. This person spikes a cookie, but not a banana. So a little more typical. We had found that some people spike to bread and peanut butter, others to protein bars, which have carbs in them. Many of them do. And 80% of people spike to cornflakes and milk, the stuff we think is quite nasty and probably not good for you. So anyway, very severe spiking there. So again, people spike to different foods. We went on and we actually spun this off. Forgot to say at the outset, I'm 100% conflicted with everything I'm showing you, but I want to show you some data from a company because I think it's interesting. We spun off a company called January AI. There are a number of companies out there, so I'm not the only one conflicted, I guess. There's Levels, there's Zoe, others that are actually, trying to use continuous glucose monitors to better manage people's health. And so what January AI did, they've run several studies. I'll show you this one where they had 1,000 participants, about 655, were doing very active food logging and lots of data. And basically what they found was just wearing an app for 10 days, or the CGM for 10 days, that's how long they were at the time. Some of them last longer now. This was done with an Abbott. The other work I was showing you was done with the Dexcom, but this, anyway, these folks found that, most people will actually improve their time in range, as it's called. Here's one example. This person, has more severe spikes. By the end, they were actually spiking less. And just by wearing the app, we weren't coaching them. We weren't telling them anything. It's very eye-opening for most people when they wear these things. And so they actually improve their time in range without us even coaching. They went on to actually build an app-based program where they track you and make food recommendations. And they have a 32 million food database, and they use a lot of AI. And the bottom line is that based on so from 4 days of logging, more is better, but a minimum 4 days of logging your food and wearing a CGM, we can make predictions about what foods spike you and what activities will suppress those spikes. And so they build personalized food recommendation programs around that for getting people to better modify their behavior.
Do you find,
know.
Mike, that there's some kind of like overarching recommendation, sort of the low carb,
Low
high
carb, obviously, yeah. There's certain obvious things to do. Go low carb. Don't eat 3 hours before bed or your glucose will be high the next day. Take a brisk walk, and they actually teach you some of the stuff in their app, believe it or not. So if you do a brisk walk for 15 to 20 minutes after eating something that spikes you, you can really suppress your spike, and we'll show you that as part of the whole thing. So yeah, there's a few standard things to do. And in the lab, we're actually doing a lot with time restricted eating right now to see its effect on overall glucose. And that's becoming popular. And there's some studies out there that say to do that, meaning try to restrict your food to within 8 hours, keeps your overall glucose level down. We certainly have found both in the lab and in the company that, don't eat for 3 hours before bed and you will keep your glucose. That helps suppress your glucose as well. Our lab's actually even gone on to show that those who do a walk after dinner actually have lower glucose. So there's a lot of simple things you can do, to your point, that turn out to be very, very powerful for keeping things down. And this group also tracks your fiber, because we think there still are fibers that will suppress your glucose. And so overall, then people who actually do this program, it actually does work. This is time in range for these healthy, and these are pre-diabetics. We didn't have enough diabetics in the study to give a proper significance here. But the trend was in the right direction. These folks will actually improve their time in range, it's called, again, for their glucose spiking. For pretty near everybody, and the magnitude depends on who you are. The other thing that was kind of cool about this whole study was that actually just going through the program with a CGM and the sort of coaching this, and it's not done with people coaches, it's done with, an app. Basically, people will they'll lose weight, and the more diabetic you are, so to speak, the more weight you will lose. So type 2 diabetics lost the most, pre-diabetics less, and normal folks still lost weight, but less. And that this is not totally surprising. It also correlates with the amount of weight they started with those. So those who are the heaviest, the highest BMI, and the heaviest lost the most weight. So actually leading these better glucose eating patterns, if you will also lead to not only improve your glucose control, but also lead to a weight loss. So the two probably go hand in hand. The other, coming back to this issue about microsampling. So we think wearables and CGM in particular are very powerful for better managing your eating, health, and overall lifestyle. We've been working a lot on this home microsampling. Actually, 7 years now we've been at this to try and find the right matrix for being able to best collect samples at home, mail them in. So that's the idea. You'll collect fixed samples at home. What we use two different devices. This is one used for pricking your fingertips. There's another you put on your back of your arm, and then you FedEx the samples to the lab, and we'll do deep metabolomics, proteomics, lipidomics, and actually targeted assays. In the end, we can measure about 2,200 different analytes in these, including some pretty high value ones. And we've done a lot of work. I'll skip over this, but most proteins we measure are stable. Most metabolites are stable as well. Lipids, some are, some aren't, but we know which ones are stable, which ones aren't when going through this procedure. And so we can get fairly quantitative measurements on a lot of different molecules. Now, it's not all the same molecules you'll measure in a physician's office. Like, we don't measure LDL and HDL, although we do measure, triglycerides and other things. But so it's complementary to what you'll get in a clinical office. And so we went on to actually show that we did two kinds of fun experiments. We showed that different people will respond differently to the same food. And the very first study we did was we had 32 people drink this Ensure shake that you can buy at CVS. And we sampled them either before, again, they can do this all at home, and they did. We sampled them before, 30 minutes, 60 minutes, 120, 240 minutes after drinking the shake. And not surprisingly, hundreds of molecules changes, from time course from 0 to 240. Red means these are the shifts, and lots of people do change their molecular composition in response to the shake. And we can track some pretty important molecules. C-peptide, this is an incretin, many of you may know, insulin, and other valuable markers. And we can also track inflammatory markers like interferon gamma. Leptin is appetite. Here's a cytokine. So we can follow a lot of pretty key molecules. One of the cool things we discovered is that everybody reacted to this food differently. So each box is a different person. And here I'm just showing 3 carbohydrates. People tend to behave the same for a particular molecular class. So this individual here, S13, their carbohydrates plummet, relatively speaking, when they drink the shake. But this person, S21's, their carbohydrates skyrocket, 24 skyrocket, 27 up, so on and so forth. So some people go up. Here's another person goes down. Here's one's kind of flat. Here's one that also goes down. So again, everybody's responding to these foods differently, and it depends on the class of molecules. Some people, their carbs go down, it's free fatty acids, other certain amino acids, and so on. And so we could group people into 5 groups, if you will. Gray is the average for all these classes of molecules across the cohort. And for this group 5, brown are the inflammatory markers. There's a series of markers following cytokines and other metabolites, if you will. And for this group of people, their inflammatory response goes down in response to the shake. And for this group of people, it actually goes up. Same with this group of people, goes up relative to the average. So same shake, some people go down, other people go up.
Mike, did you investigate, sort of differences by, race, sex, age, those types of things in terms of like these responses?
Great question. Right now, we're underpowered for all that. So we want to, now that we've seen, this is fairly new, we just described this, we're rolling out a bigger study. We're very keen on getting at some of the things you just described, but also trying to get at underlying causes. My own guess is the microbiome and transporters and things in the intestine are probably all contributing to this differential response. And so there's a lot of detailed molecular characterization to do to try and understand what's causing this. And then how it correlates with age and as you say, ethnicity is something completely unknown yet. So right now, this is mostly a European, not 100%, mostly a European cohort. Yeah, okay, great question though.
And then just to circle back to your collection, your blood spot collection,
Mhm.
how is the compliance and, the return rate of those samples? And just sort of like, I know this is happening in the T1D space. People are trying to collect data or from samples out is sort of in the field or from people's homes. And I wondered how your process is performed.
Yeah, great question. Believe it or not, we're running nearly all of our studies this way now because it's easy to do, it's cheaper, and you're doing it at home where people are in a more natural setting. So we are running a lot of studies. For this study, the compliance is nearly 100% because they, they only had to collect 4 samples over, up to 4 hours. So it's pretty easy for them to collect all the samples, mail them all in. And so compliance was really, really good. For some of the other studies we're running, like we're running some mental health where we're following people out to a year, it's still pretty good. I'd say we miss from those who start, we might miss about 25% of samples at any one time point, at subsequent time points. But we do keep on top of people. We have to ping them to say, don't forget to send your samples if they haven't done so already. We remind them ahead of time, oh, it's time for your sampling. And then if they don't send it, we try and ping them to keep it in. So the compliance overall is roughly, I'd say about 75%.
Okay.
It is pretty easy to do. And that would be for a mental health study where, some people, I guess, get disinterested perhaps more than some of the other studies. We're doing it for glucose control studies. Those folks are probably more compliant. It's a great question, though.
And then what is the payer situation for the processing of those samples and the kits? Does that come directly out of your grants?
It is, yeah. So we run the study. We don't pay participants. We like to give them the results of the study so they can see, what came out of this. So for these particular studies, we're not paying folks. We have some other studies. We're running one, a pain study actually, where just they're doing a lot more. So that particular study, they're getting paid. But for most of microsampling, we just they just do it. We mail them the kits and we give them a FedEx envelope and they mail it back.
That's pretty good. And then from the chat, someone's asking, how do you assure that the measurement timing is consistent in relation to consumption?
Yeah, it is self-reporting. We try to give them very specific instructions. I think in this case, it's not too hard because, and so could people be shifted a little bit? Probably, but we asked them to record when they actually took the sample. So we do try to get that information, and certainly as they've written it and sent it back to us, it does, it complies pretty closely with what we've asked them to do. In the case of some of these studies, like I mentioned, this mental health study, they'll be within a few days. We always track the date by which they send back samples. So we are following all that. And I should say, I didn't show you the slide, but where we did all the stability measurements for the analytes, we actually put them at different temperatures for different lengths of time. So we really know which ones are really stable and we're getting pretty accurate measurements on. As I say, a few of the analytes, we don't get the measurements aren't as good. We leave them out of our analysis. Okay. So the other thing I wanted to tell you, this is where I see this going. This is the ultimate, we think, in health monitoring, where this is one individual shows you what you can do. They took samples for basically every hour over 7 days. And every waking hour, not at night. And they're wearing a CGM and a smartwatch and doing food logging. So you could track food that was eaten with activity and physiology and biochemical responses. So you can make very, very detailed correlations on people. And we do something called time lag correlation, where we're trying to track what events occur upstream of other events. They're more likely to be causal. And from this kind of analysis, we actually find thousands of associations between glucose, heart rate, and steps and biochemical association. And we can quantify precisely in a person exactly how quickly their C-peptide, their incretins go up after eating and that sort of thing. So it turns out to be it's quite powerful. One of the surprises was we found this protein, alpha-synuclein, which has been implicated in dementia and Parkinson's. It actually has a very interesting pattern. We think it's responding to stresses. We're still trying to sort out what stresses will trigger this. Whoops. But the cool thing is that, yeah, alpha-synuclein, we think, and it does have a correlation even with glucose. Yes, it's a stress responder. So we think that people who might be, high risk for Parkinson's or dementia, it might pay for them to actually track this. And so this is something we're doing as a research and then maybe later as a clinical assay to be able to track this so that we could, again, try to have people keep their levels lower, which should lead to reduced, dementia and Parkinson's. That's the hypothesis anyway. So these are the kinds of things you can just do and discover when you're doing these deep profiles.
Yeah, and just as sort of a plug for type 1 diabetes, low alpha-synuclein levels in the blood are associated with type 1 diabetes.
Oh, is that so? Low levels. Oh, that's interesting to know. Thanks for sharing that. Great.
Yeah, I'll drop the paper in the chat.
Cool. Love to see it. So I'm totally conflicted, as I mentioned before, but one thing we do, our lab is a research lab, but we do spin off companies, and I've spun off 17 over the years of different sorts, so genome sequencing and other sorts. And so we decided that we thought this was pretty powerful. We're in a world now where metabolite information, we think is very, very powerful. Obviously, this group's very sensitive to glucose, but we think other metabolites have a lot of value as well. So we've set up a home sampling test, so relating a little bit to what you were getting at earlier, Monica, where you actually can, you visit the site, give a sample, and they will give you back, they'll analyze 500 plus analytes, and they're associated with about 20, if you will, wellness categories. And the information we think is actionable, as I'll show you in a minute. So as an example, this is, these are just showing 6 health categories where you can track, metabolic health, inflammation, oxidative stress markers, and see how they're tracking bone health and other. We even follow longevity because we can classify, because we know the various metabolites versus age, we can basically see where you sit in your age categories and set up, if you will, aging markers. And in fact, to push this a little bit further, so we'll get back a health report, but we can get back an aging report. This person happens, their chronological age is 30, almost 32. Their biological age is pretty good. They're 22. Here's a more typical scenario. This person, their chronological age is almost 51. Their biological age is 53. So they're a little bit older biologically than their actual age in years. And the cool thing, it's like the age of types. You can see what the subcategories are that are contributing to that. And so this person, their heart age is older, their metabolic age is a touch older, and their inflammation is actually younger. So you could actually work on various things that might improve your heart age, if you will. And so we actually have we'll summarize the findings, and then we use AI to actually go into our database as well as literature that we've curated. And we can pull back and give recommendations that we think should better, for lifestyle recommendations that we think should better improve people's health. So, for example, once you get certain things back, well, they might recommend taking some of these fibers. It also integrates, I should say, your medicine and lifestyle information, foods you eat, cut back sweets, add more polyphenols that should improve certain of the markers. And so again, it's actually been pretty powerful. And most people do act on it. Turns out 95% of people who get this information do improve their markers and then after getting the information. So this person, for example, improved this particular marker by following we gave them the recommendations. We think they followed And so basically we think this is powerful. You notice some branched chain amino acids here. So again, 95% of people did improve after their first visit, their, if you will, metabolic markers. A lot of these markers, they're on the FDA site. They're not validated clinical tests, but they are FDA informed markers, if you will. So overall, what I've been telling you about is that we all respond differently to different foods, and metabolic markers, inflammatory markers, all will shift differently in response to different foods. And I showed you examples of fiber, the Ensure shake, glucose response to different foods. I mentioned that overall rhabdoxylan is good for cholesterol. We have a whole new mechanism for how we think that actually happens. It's a little different than what have been put out there. I mentioned that different people spike their glucose at different foods, and you can actually, CGM, we think is a powerful way to improve all this, and there now. The idea that you can scale this, and we think this is a big deal because I'm sure this group appreciates 9% of the US is type 2 diabetic, and 33% are pre-diabetic, and those numbers are going up and up. And so we think this is a scalable solution to try and at least help people improve their glucose dysregulation, which you think will be quite powerful. And then I mentioned how metabolomics provides actual markers into wellness and aging. And so, yeah, I guess those are major conclusions. These are some of the folks, I put these up, you know, again, I'm conflicted, but some people ask me, so if you want to pursue it further, by all means, you know, take a picture of this, and I guess you display the webinar later. But Sam led and Brittany led the fiber work. The CGM work was started originally by Heather Hall and Dolly and Alessandra. We have great collaborators with Justin Sonnenberg and Tracy McLaughlin. And then our wearables work has been pushed out by many folks, Xiao Li and others. And the microsampling has really been spearheaded by Xiao Tao and Ryan. So that's the story here. Happy to take questions if you have any. I guess I can leave this up if you want to copy any of this. Whoops, any of this information. You know, let's stop sharing here. Maybe that's best. So yeah, happy to take questions. Let's see if there's any in the chat. You've been asking some.
Thank you. Yeah, thank you, Mike. This is great. This has been really great. I just wanted to, I'll just paste this in here for that. And it's actually the alpha-sin is highest in kids with obesity, followed by kids in T1D. So it's a little bit lower than those in obesity, but it's not low est. Let's say that.
Yeah, that might fit then with the idea of the stress triggering this. That's interesting. Yeah. Okay.
And so here's a question. Can you share some pearls, lessons learned regarding big data collection analysis for the T1D research community? Including on remote collection?
Great question. So unfortunately, we haven't done as much on type 1 as we have on type 2. So sorry about that. But we are doing a lot of autoimmune in general, mostly in the area of rheumatoid arthritis. So we can show from these microsampling that I mentioned devices before that you can actually follow people's autoantibodies and such. You can elute them off and then run Luminex assays to be able to follow both cytokines and growth factors, all kinds of hormones. So I think it will be very powerful. I think for those of you who are on certain medications, most people are obviously insulin, but I think you could track this. And I showed before that you can track C-peptide and insulin pretty well. And you should be able to follow your exact kinetics, if you will, by actually using this approach. Right now, as I say, it's all research, but at some point it will become mainstream. Now, my collaborator, Tracy McLaughlin, has a type 1 study running, not with microsampling so much, but very interested in CGM, still trying to improve glucose control, even for type 1 diabetics. So, yeah, but.
Is she at Stanford as well?
She is, yeah.
Mm-hmm.
So, but maybe it's just because of my particular situation. I am type 2 diabetic. It was predicted from my genome and I actually got it after a nasty respiratory syncytial virus infection of all things, before COVID. So my thought is I'm genetically predisposed and the viral infection triggered. It's not so unlike what's thought to happen perhaps in type 1 diabetes, as I'm sure
Right.
you know. It's generally thought, although it's not 100% clear what the triggers are in type 1. That's what the TEDdy study, many of you may know, is all about. They're trying to figure out the triggers, and we should be measuring the exact viruses and pathogens that are affecting people, because that's when, well, type 1 and type 2 is now appearing, or so
Yeah.
it's.
Well, I mean, the thought from listening to the field, currently there's a variety of endotypes and paths to become type 1 diabetic. So that's
Right.
definitely sort of un
But yeah,
understanding these paths is very important.
and they
And I
may all
would
have
also,
stress in common, right, Monica?
yeah,
So that's the common element, perhaps. Yeah. But then I think
yeah.
you want to know which ones are more than others. I'm sorry, I cut you off. Keep
No,
going.
no, that's true. And I would also say, in terms of C-peptide, we have a sort of internal discussion about C-peptide and how it's not totally standardized across communities. So that is also something that needs to be
Yeah.
shored up. And then I would say there's a couple more questions here. Do you have any this is coming from England, do you have any multi-omics data, or actually from Wales? Do you have any multi-omics data from people with hypothyroidism, particularly those who still report poor QOL and LT4?
So the answer is yes. We have one person who is reduced in making certain, and we have another person who has some. That's more common than you think. Some people with hyper thyroid hormone. So we do have some of those, but I don't know in terms of what other, what pathways are elevated in those folks. But you could actually, all our data, by the way, what's pretty cool about that original study I mentioned of the 109 people is that they've agreed to make, or most of them, all their data completely available, including their genome sequences. So you can actually, and my understanding is a lot of you are scientists, so feel free to download and explore and play with those data. And for the few who aren't making their genome sequence data available, all the other data are available. And
Where are those data housed, Mike?
yeah, so you should probably write to me. They're in our Stanford data ocean, and I can steer you, or you can find their papers, and they will steer you to a site, and they're generally in the same general database. So feel free to either reach out to me or look at some of our papers, and then love it if you start exploring it. If for some reason, the links aren't working, which does occasionally happen, we try to keep them active. Do let me know, and we will get you the data. So we're very happy to have it explored. It turned out to be a very valuable resource. And CGM, the first study is called IPOP. That's all kind of in one site. The CGM data, it's still in the same general database, but it's a different study, so it's different. We're one of the few groups that shares the CGM and the wearable data. You can download it directly, no permissions needed, etc. So,
That's
um,
great.
yeah,
Really, really collaborative, and I applaud that.
thanks.
And then here's another from Bridget Pronert. Have you looked at differences in response to food / types of carbs in people with type 1 diabetes? An example, looking at response independent of insulin secretion or
Hmm.
response with a set insulin dose.
So the answer is no, and we haven't done that even for type 2. We're just, basically characterizing the phenomenon. What foods spike what people? Again, all pretty type 2 focused, though. Although some folks may be heading towards, they're starting to take insulin, so there may be more insulin dependent type 2s rather than type 1 per se. So I think you're asking a great question. And I'll tell you, there's a lot of fun data in here, and it'll be fun to do this for type 1. We are interested in that side on the food responses. And if I were to show you the data, for example, berries, you'll get a big spike if you eat berries, but it comes down really rapidly. And if you eat rice or potatoes, you'll also get a big response, about the same size, but it doesn't come rapidly. It stays up for a while. So the area under the curve for potatoes is actually quite large. So we think these different responses are a big deal because I think they're actually reflecting both the food and how you're handling it. And by the way, that also I told you what was generally the case, but for some people, it's different from other people. And so I think it reflects the underlying glucose dysregulation they have. I mentioned before about type 2 diabetics being, different subtypes. And we think that's why people are responding differently.
Yeah, and then Bridget Fronert also said anecdotally, patients report different, sort of in quotes, trigger foods, which are associated with higher peaks. So this is such a
Yep.
really interesting, foray. And I mean, places like Tide Pool, Dexcom, they have a wealth of these data sets. So it would be great if, scientists started to wade in and more fully understand the type 1 diabetes cohorts in context of what you're doing here. Just one last thing, you've got this Stanford Data Ocean. I know they have SURF as well. How accessible are those? This is a scientific audience, so how accessible are those, data sets sort of overall to other scientists? Again, is it just sort of a matter of
100%
reach?
open. 100% open.
That's
So
great.
yeah, and that's special. We only enroll people early on. We let anyone enroll. So there's a few where they don't share their genome sequence, but they have to share everything else. But as say 85% of people share everything, including their genome sequence. So you'll be able to download. Same with the CGM, the wearable data. It's all available. And so as I say, sometimes people have trouble getting things in the right format, and we have somebody online who can help you with that, who will steer you if you have problems. Most people who are pretty comfortable with dealing with big data sets know what to do. But if you have any issues, we have someone who will help you.
A bioinformatician. That's fantastic. That's
Yeah, we
wonderful.
think it's a big deal because I've been through this myself, for example, where you'll see in papers where people say, well, data available upon request. And we always request it. We never get it. It's actually a problem for both wearable data and CGM data. To the best of my knowledge, there's almost no public data out there for that, which is a little annoying because most patients want to share their data, is our
Right.
experience.
Yeah,
It's,
we understand that very
yeah.
well. We actually initiated and facilitated the build of a T1D TCR BCR
Oh,
genetic data base, and it's building. But we did that because we realized that a lot of these data sets are siloed and inaccessible, and we wanted to open it up to the T1D scientific community.
right. Yeah.
So that's great that Stanford's taking the lead in this. It's really, really great to see.
Yeah, well, as I say, we're not as embedded in type 1. Sorry, folks, but ask me
Well,
to
maybe you will be after this
back in
talk.
2 years. Yeah,
Yeah,
yeah.
maybe
So,
today someone one of your trainees will get the bug
right.
and start looking.
And we
Uh,
are playing a little bit with that, but not at the level of type 2, but we will.
it's great. Well, thank you for all you're doing and for offering these insights and accessibility to these data. It's really great. And hopefully it'll inspire someone who listens to this or watches this and to reach out and get more traction. So thank you again, Mike.
Thanks for having me. Bye all.
Great. Bye-bye now.